My name is Jason Amsbaugh, and I'll be the moderator for the webinar today. Our feature today is Dr. Dirk Hockemeyer from UC Berkeley. He leverages his expertise in telomere biology, mouse genetics, human stem cell techniques, and genome editing to discover the mechanisms of telomerase regulation in human stem cells, particularly during tumor formation. We will begin the webinar with Dr. Corey Padilla from Dovetail Genomics, followed by Dr. Hockemeyer, who will talk about recent discoveries in human cancer samples that have uncovered mutations that influence telomere length dynamics. This work has led to investigating the role of telomere-telomere fusions and the contribution of dicentric chromosomes in genome evolution, which is some of what he will highlight today. So without further delay, I'll turn the time over to Dr. Corey Padilla.
Thank you very much for that intro, and I'm just going to give a very brief introduction to what we do here at Dovetail Genomics, and then pass it off to the main event. We don't want to keep you too long for that. So just as a very brief primer here, most approaches to NGS-based genomics result in a linear view of the genome. So here we're showing varying genomic variants on a reference genome, and you sort of see how they're all sort of visualized in a linear space. This also goes for gene regulation, where you see your core promoter and the regulatory elements that sit some distance away, again in linear space. But we know that's not really the case, right? Genomes are quite long in the human genome. That's 3 meters long, and each cell has to package that 3-meter molecule into the nucleus. And so to do that, there's a lot of folding that occurs, and this results in a hierarchical structure of genome folding that has implications on gene regulation for how regulatory elements like enhancers that can be many kilobases away interact with their target promoter to kick off gene transcription. So we here at Dovetail have a handful of options for people to look at those chromatin interactions in 3-dimensional space, from our OmniSeq kit, which enables variant detection from SNPs to large structural variants. It does a really good job with genome assembly and phasing across the whole chromosome. This is also compatible with hybrid capture approaches with existing panels. You don't have to go in and redesign anything that you're looking at. We also have a nucleosome-centric view of chromatin topology, so you can really dial in your questions to the nucleosome resolution. And then we have a chromatin immunoprecipitation approach, which really allows people to target chromatin interactions with a protein DNA interaction of interest. And so with that, I just want to say, thank you for attending today's webinar, and we're really excited to hear the following presentation.
Thank you, Corey. We'll go ahead and turn our time over to Dr. Hockemeyer now.
Good morning, everybody. This is the first time that I'm giving a webinar. Thank you for coming to this little experiment that we're doing. I hope you're all well. This is a great opportunity for me to, during these really trying times, to enjoy research and presenting. I have sort of a two-phase talk today, where the first part of my presentation will give you a little bit of an introduction what my lab is doing and why I'm interested in chromosome biology. The picture that you currently see is a fish standing for the end of chromosomes, which will be the main theme of my talk today, telomeres. And then in the second part of my presentation, I will venture into some exploratory unpublished work that we have been doing together with Dovetail Genomics. And I think this is a good marriage to show some of the outreach experiments that we are doing in my lab to understand chromosome biology, implementing new technologies. The center of my lab is understanding stem cell biology. I'm particularly fascinated by stem cells. As you know, stem cells are characterized by the main feature of being able to differentiate into somatic cells, but also to self-renew and regenerate themselves. And so this difference in the human body that there are stem cells and somatic cells is actually a key feature of human biology, because it allows to separate two main functions. That is that the somatic cells carry most of the burden of the proliferative burden that execute the function of the cells, while the stem cells replenish the somatic cells and allow for the rejuvenation of tissues. This division of labor allowed the human body to develop a molecular clock that curtails the proliferation of the somatic cells, limiting the amount of divisions that cells undergo. And this functions as a strong tumor suppressor mechanism, because if you limit the proliferative capacity of somatic cells, then this prohibits the expansion of this population towards cancer. So this division of labor functions as a tumor suppressor mechanism. On the other hand, this molecular clock that has evolved is necessary to support the function of the stem cell to allow for self-renewal. So this is a really interesting balance that has to be struck by the stem cell, and it's a function of cellular biology that extends over many years of the human body. If you think about it, the stem cell in our bodies are able to sustain tissues for almost 100 years. So the balance between allowing the self-renewal capacity of stem cells and somatic cells that they are curtailed in their ability to proliferate is a key function. And so if this molecular clock breaks, then this leads to tumor progression in the somatic cells, while if this molecular clock breaks, it suppresses the self-renewal capacity of stem cells and thereby leads to tissue failure or aging syndromes. So the central statement in the central feature of this molecular clock is hardwired in the structure at the linear chromosomes, which I will introduce to you today, which are the telomeres. And so my lab is interested in understanding the molecular features of this clock and thereby understanding how pathologies are driven by the dysfunction of this mechanism. The general hypothesis is, since these biological processes extend over very long periods of time, we are studying the extremes of when this clock breaks. So pathologies that are involved in mutations in this machinery that lead either to cancer or to tissue failure syndromes, we are studying in order to extrapolate how the machinery works in order to allow long-term stem cell function and tumor suppression. So what I would like to do today is I will first briefly introduce to you the key features of how telomere biology sets this molecular pacemaker for cellular divisions. Then I would like to introduce to you how we study mutations in telomere biology that are involved in cancer formation. And then I would like to quickly tell you about some of the experiments that come out of this study of cancer biology, how we use Hi-C methods to study structural variants in cancer. Okay. So this is another fish image of telomeres, which are the linear ends of chromosomes. They are comprised of TTA GGG repeats. These repeats are, are bound by a protein complex that is referred to as the sheltering complex that has 6 protein members. And these buffer sequences at the end of chromosomes have 2 main functions in cells. The first one is that they are dispensable sequences. So that when the chromosome end shortens due to the end replication problem or through nucleolytic degradation, that the sequence that is lost is non-coding and dispensable. The second function that telomeres have is that they make the natural chromosome end distinguishable from internal chromosome breaks that have to be repaired, while the natural chromosome end is not allowed to be subjected to this repair mechanism, because that would lead to chromosome fusions. And that will become one part of the things that we will discuss today. Telomeres can be maintained by this enzyme telomerase that can de novo synthesize these repeats. It has an RNA subunit and it has a protein component that is called Tert. This Tert protein, the catalytic subunit of telomerase, is one of the proteins that I will discuss in detail today because it sets up one of the key features of how the molecular clock works. In human cells, this protein component Tert is transcriptionally regulated so that it is only expressed in stem cells, but that most somatic cells transcriptionally silence the expression of Tert. This leads to a very striking difference between stem cells and somatic cells, which is that in stem cells, the expression of Tert leads to the maintenance of telomere length over time. Telomeres do not shorten in human stem cells, while in somatic cells, telomeres progressively undergo repeat loss at the end of chromosomes. We generally assay this telomere shortening by extracting genomic DNA from cells, cutting it with frequent cutters that do not cut in the telomeric sequence, but cut the rest of the genome so that the telomeres stay intact. And then if we resolve them on an agarose gel and probe them with the telomeric sequence, you can appreciate that the terminal restriction fragment that can be hybridized with the telomeric probe progressively undergoes shortening. You can appreciate that telomeres are different in length, which leads to this SMEE. This is the image that you get if you perform this assay in human fibroblasts. This progressive telomere shortening has a dramatic effect on cells because it sets up this proliferative capacity that is referred to as the Hayflick limit. Cells that undergo telomere shortening enter a state that is called replicative senescence, where now telomeres become detected as sites of DNA damage, which can be illustrated by the localization of DNA damage factors to telomeric sites. This is a staining for the telomeric binding protein TRF1, and you can appreciate that the localization of DNA damage proteins in this set up a DNA damage signal that leads to senescent phenotypes, which can be shown here by this beta-galactosidase stain. This is a very strong tumor suppressor mechanism that is set up by a p53 response in cells that are impaired in their p53 and RB response, for example, through the expression of SV40 large T. Chromosomes can continue to divide and leading to further telomere shortening. And at this point now, telomeres not only sense as sites of DNA damage, but also are detected as DNA damage foci and are repaired, leading to chromosome fusions, which can be highlighted here by this dicentric chromosomes that, as described by Barbara McClintock, go through breakage-switch fusion cycles and further genomic instability. 85% of cancer cells overcome this proliferative barrier and this genomic instability by expressing the protein component, by being telomerase positive and by reactivating the expression of the protein Tert that led to this initial telomere shortening. And what you can appreciate is that in tumor cells now, the telomeric restriction fragment is stabilized, and over consecutive division, telomeres do not progressively shorten anymore. And the reactivation of telomerase leads to in the immortal phenotype that we find in tumor cells. This is a really interesting event, the reactivation of telomerase. And when I started my lab, we were particularly interested in understanding the mechanism of how this reactivation works. And we were quite lucky. When I started my lab in 2013 in UC Berkeley, simultaneous with the start of our lab, there were 3 papers that identified mutations in the promoter of this protein Tert that are single nucleotide mutations, which turn out to be the most frequent non-coding mutations in human cancer. They have an overall prevalence of 10 to 15%, maybe even higher than 15% of all cancers carry one of these mutations. These mutations lead to X factor binding sites, and they are found with a very specific pattern and cancer spectrum. They're almost never found in leukemias, intestinal cancer, or breast cancer, but have a very high prevalence in HCC, BCC, melanoma, and glioblastoma. So this was a smoking gun, and we decided to study these mutations and investigate their role in how they promote cancer biology. And so we wanted to ask two very simple questions. The first one is, what is the mechanism of how these mutations work? And then the second question that we had was, how do these mutations occur in such a specific cancer pattern? And so two of my students, Konitoshi Chiba and Francisca Lober, addressed this question. And we did this by turning to our favorite model system, human embryonic stem cells, and decided to engineer these mutations using CRISPR-Cas9 and generating an isogenic panel of human pluripotent stem cells that differ exclusively at the individual cancer mutation. This was actually a really heroic feat of engineering these mutations. The Tert promoter is extremely highly GC rich, and when Chiba started making these cells, he came up with a really elegant way of making these mutations, and that's summarized in these initial slides. The way that he decided to make these mutations is to use an editing approach where he used two Cas9 cuts that delete the entire Tert promoter. These Cas9s were in regions that were not highly GC rich, so that he could make a homozygous cell line that had this part of the Tert promoter cut out. This leads to a cell line that we refer to as Tert delta delta. And if you assay for the activity of telomerase in these cells, which you can do by asking for an oligo being extended by telomere in vitro, extended for TTA GGG repeats, this is one repeat added by telomerase 2, 3 repeats. You can see that this Tert delta delta, because it cuts out the ATG of the Tert gene, are telomerase deficient. Since they are telomerase deficient, these cells will undergo telomere shortening and eventually die. Unless you reopen this cut in the cell line that is on its way to die because of telomere shortening, and complement this phenotype by inserting the wild-type promoter back into these cells. And so when you complement these cells with the wild-type promoter, these cells are now allowed to live. If you do the same experiment with donor plasmid that contains the mutation, you can generate isogenic cell lines that differ exclusively in the mutation status of the Tert promoter. And so when Chiba made these cell lines for all three Tert promoter mutations, he was super excited. It was a significant amount of editing that he did. And then he asked, what happens to telomerase activity in these human pluripotent stem cells? And that is summarized here. This is, again, this telomerase activity assay for wild-type cells or human ES cells that have these mutations. And what you can appreciate is that there was not much of a difference. And so that was, on first sight, quite frustrating because, of course, we hypothesized that these mutations would lead to a difference in the telomerase activity in these cells. So I have to say that at this point, there was a lot of work that went into this, and we were a little bit scratching our heads. But Chiba is an extremely smart guy, and he said, Dirk, did we do the right experiment? Because we are hypothesizing that the mutation breaks the tumor suppressor mechanism in somatic cells, but we are assaying the cell mutation in a stem cell that is already immortal and can self-renew. So wouldn't it be the right way to test these mutations in actual somatic cells? And so he went on and tested the hypothesis that he formulated by taking these Tert promoter mutations in human embryonic stem cells and differentiated them into somatic fibroblasts that are naturally telomerase negative. And that's where the story started to become really interesting. This is the expression of Tert in human embryonic stem cells. If you take these cells and differentiate them into fibroblasts, Tert expression is silenced. However, if you do this differentiation in cells that carry any of these Tert promoter mutations, we see significant increase in residual Tert expression. And we see that the silencing of telomerase activity, which you can see by this loss of extension of this radioactive product, is also abrogated by these Tert promoter mutations, indicating that the key defect of these Tert promoter mutations is an incomplete repression of Tert activity, and it's not necessarily an activation of the Tert expression. So with this, I think we had already insight into one of the key features of these mutations by highlighting that a Tert promoter mutation in a cell that is normally telomerase positive, like a stem cell that is giving rise to colon cancer or to lymphoma, might be neutral and not be positive selected because this cell is already immortal, has a telomerase, expression. And it highlights that the mutation only will exert its tumor promoting effect in a cell that actually undergoes telomere shortening, that turns telomerase off, and in a compartment where the proliferation is actually restricted by telomere shortening, such as a tumor initiating cell in melanoma or hepatocellular carcinoma. So we were quite happy that we could answer the first question that we set out to ask, but there were two things that arose during the time that we were doing these experiments that we were wondering about. And that is, I introduced to you this very simplistic model that telomeres shorten. And then at this point where telomeres become critically short, they sense a DNA damage signal. And that is when a cell comes up with the Tert promoter mutations and stabilizes their telomeres. What argues for this, what argues for this model is that telomeres in cancers that have these Tert promoter mutations are short. And that is coming out of cancer genome studies. And what supports this model is that cancers with TPMs have an increase in genomic instability. However, what argues against this idea that Tert promoter mutations are derived during crisis is that in melanoma, at least, we knew from also cancer genome sequencing studies that Tert promoter mutations can occur and are positively selected before the loss of cell cycle checkpoints, such as loss of p53. The other thing that argues against this very simplistic model here is that short telomeres are protective, particularly in for example, melanoma. And that argues against that the frequency with which cells enter crisis should be a positive selector for the indication of Tert promoter mutations. So to reconcile this seemingly simplistic model, we decided to use our cell system and now push this a little bit further and ask what happens to the cells that we actually differentiated into fibroblasts, and do these cells truly immortalize? What happens to telomere length in these cells? And what happens to genomic instability in the cell system? So we took our panel of isogenic fibroblasts and we first addressed immortalization. This is the result that you get if you take human fibroblasts that have one of these Tert promoter mutations and compare their proliferative capacity to isogenic wild-type cells. And I still think that this is one of the really coolest results that my lab had so far. It's a very simplistic assay. but it highlights that wild-type cells that undergo the Hayflick limit stop proliferation. This is the tumor suppressor mechanism that I mentioned earlier. However, a single point mutation in the human genome allows for the indefinite proliferation of a cell over time. And that, I think, is a really cool result, arguing that these mutations are truly sufficient to immortalize cells. Next, we ask, okay, so this argues that the problem is solved, right? Telomere biology is not an issue in these cells anymore. Telomeres are expected to not be a problem. However, Chiba decided to actually verify that and to test what happens to telomeres in the cell system. And that's where he made another really important and surprising discovery. This is the telomere shortening that you see over time in wild-type fibroblasts differentiated from human cells. You can appreciate that telomeres progressively shorten. However, if you ask for what happens in cells that carry the Tert promoter mutations, you get this result. You can see that also in cells that are seemingly immortal, based on the data that I just showed you, telomeres also initially progressively shorten and only stabilize at a very late time during their proliferation. And that seems to be a contradiction. That seems to be at least surprising. And we were, again, scratching our head and we were thinking, how do we reconcile that? Before I resolve this, we were so surprised by this result that we decided we want to confirm this in an actual cancer-relevant system. And the way that we did this is we collaborated with Boris Bastian at UCSF. Boris Bastian and his group have a very intriguing cell system, a melanoma system, that allows them to study the progression of melanoma development. He has cases where he identifies nevi that are adjacent to a melanoma, where the nevus is the precursor state for the adjacent melanoma. And he can excise these lesions and then sequence the mutations that led to the proliferation of the nevus, which are usually driver mutations in the MAP kinase signaling, and then the progression to the melanoma. And what he identified is cases where the Tert promoter mutations were the next mutations at the transition from nevus to melanoma. They are found in the melanoma, but not in the nevus. And so using the system, we used fluorescent in situ hybridization to ask what happens to telomere length in this transition cases. And again, we find the same thing that nevi that are wild type for the Tert promoter have longer telomeres than the adjacent melanoma, arguing that at the transition from a nevus to melanoma, the occurrence of a Tert promoter mutations did not halt telomere shortening, and that telomeres were allowed to progressively shorten until they ultimately stabilized. So with this corroboration of that what we observed in our stem cell system actually is recapitulated in melanoma samples, we came to the conclusion that a Tert promoter mutations is not sufficient to halt telomere shortening. The only way that this can be reconciled is by arguing that telomerase initially is not sufficient to stop telomere shortening. This is a depiction of a condition where telomerase is abundant and highly abundant. And what we know under those conditions is that telomerase, when it is present in excess, will not elongate all telomeres at all time, but will find preferentially the shortest telomere and only elongate the shortest telomere. However, if you have limiting amounts of telomerase, and this is what happens when you differentiate cells, telomerase will again only find the shortest telomeres And under those conditions, the longest telomeres will allow to progressively shorten. If you have critically levels of telomerase, then the telomerase present in the cell is only sufficient to find the short telomeres, and thereby telomeres in the population over time continuously shorten, and telomerase will only attend to the short telomeres. So if this was the case in our Tert promoter mutation scenario, then this makes two predictions. That eventually this process cannot go on forever, and that cells have to make two choices. Either they will die because the telomerase driven in genomic instability and telomere fusions will eliminate the cell, or the cell has to increase secondarily telomerase expression by upregulating telomerase levels over time in cells that have Tert promoter mutations. And so we tested this first. We asked for what happens in cells that have the Tert promoter mutations over time in terms of telomerase levels. As you can see here, in wild-type cells, telomerase expression or telomerase activity is completely absent in fibroblasts. And this is also true in very low detection of telomerase in any of these isogenic cells initially after differentiation. However, over time, when telomeres continue to shorten, we find that telomerase levels stabilize and increase in these cells, specifically in the cell lines that have these Tert promoter mutations, arguing that the Tert promoter mutation is only permissive for the upregulation of telomerase to levels that are immortalizing cells, but that initially they do not grant cells the telomerase activity that they eventually need to acquire. So these Tert promoter mutations are sort of an epigenetic lesion that allows for the progressive upregulation of telomerase levels over time. We tested the second hypothesis that I outlined here, and that is that some cells that do not do this upregulation of telomerase will undergo telomere-telomere fusions. And I introduced this to you earlier. This is a dicentric chromosome that is generated by having deep protected telomeres. You can assay for the presence of these dicentric chromosomes by using a PCR-based approach where you have PCR primers that are facing outwards on individual chromosome arms. Here we are assaying for telomere fusions in the subtelomeres of the X and chromosome 17. And what you can appreciate here is if we follow these amplicons over time, we see that there are very few telomere-telomere fusions initially when telomeres are long. However, over time in this bulk population of fibroblasts, in cells that in this case have this 146 Tert promoter mutations, we see the accumulation of these Tert promoter mutations, arguing that Tert promoter mutations do not fully protect all of the cells present in the population, and that some undergo telomere shortening and genomic induced instability. So that allows us to refine our model of how Tert promoter mutations change telomerase levels over time. Instead of leading to an immediate telomerase level that is cutting the threshold that allows the maintenance of all chromosome ends, we hypothesized that the Tert promoter mutations lead to telomerase levels that will gradually increase, and that this gradual increase is largely driven by a time where telomeres are undergoing or are in a marginal telomere length, just critically long, selecting for the cells that just are able to maintain telomeres over time. And this selective pressure leads to the slow and progressive upregulation of telomerase levels until telomerase levels reaches a threshold that all telomeres can be maintained over time. So I find this highly interesting because, as I mentioned, we validate that something like this has to happen during cancer progression. And here I show you the progression of melanoma that go through individual steps of cancer development. And we know that Tert promoter mutations occur somewhere during this transition of cancer development. And on the human skin, of course, this takes quite a long time to develop. And so Tert promoter mutations are prevalent in melanoma, but they're found in many other cancers. So the really open questions that I think are really interesting is in where and in which cancers is telomere shortening relevant for these progression? When do these Tert promoter mutations occur? What are the cellular stages at which Tert promoter mutations are selected for? And how does it work for cancers that we don't see and where we can't profile this because they are happening at so early stages that they are still very, very small? So to better understand the selective pressures and the dynamics of these early development of cancer, we wanted to know, can we actually detect telomere-telomere fusions in early primary cancer samples? And that is the segue into our collaboration with Dovetail, where we got interested in how do we find large structural variants? How do we understand chromosome evolution? How do we understand the dynamics of chromosome interactions in very, very small samples? And that is the transition into the second part of my talk, where we became interested in this question. And in order to explain how we got into this collaboration, I have to introduce to you my friend and colleague, Dan Rockshar, who is in the same department here at UC Berkeley as I am. And I talked to him about our studies in melanoma development, and I talked to him about our Tert promoter mutation story, and discussed with him how do we understand the genome of cancers. And Dan is an expert in genome assembly, and he mentioned to me something that I sort of knew, but and learned during about you in college, but that I completely forgot. And that is that there is actually telomere fusions present in biology that are really interesting. And that is the chromosome evolution of the Chinese muntjac and the Indian muntjac. And this is one of the most fascinating chromosome evolution that is out there. The Chinese muntjac has an N of 23 chromosomes. So very similar to our human genome. However, there is a related species of the Indian muntjac that has 3 chromosomes, an N of 3 chromosomes. And these are species that are evolutionary only about 5 million years apart. And even more fascinating is that these can make an F1. They can have live offspring that in itself then is not fertile, but it can make live offspring that is viable, although they have so largely divergent chromosome numbers. So discussing this with Dan, we said, isn't this one of the best examples where we can actually study and understand how to detect telomere-telomere fusions? How do we detect the evolution of the condensation of chromosomes from an ancestor that gave rise to a species that had 23, an N of 23 chromosome and one that has of 3. And so we decided to do that. And this is where Dovetail Genomics came into, because the platform that we use to assemble the Chinese and the Indian muntjac was the Hi-C technology developed by Dovetail Genomics. Before I show you the data, I have to mention that this is not the first time that telomere biology and muntjac have been connected with each other. Of course, Woody Wright and Jerry Shea 20 years ago already looked at this and characterized the telomere biology in some of the muntjac cell lines. And I want to give a shout out here to Jerry and Woody who shared the cells that we sequenced in this project with me. Okay, so the idea was we took cell lines from the Chinese and the Indian muntjac, and then decided to use the Hi-C sequencing platform from Dovetail Genomics. We generated very high quality genome assemblies using largely this platform, which is based on proximity ligation. Then you cut the genome with a frequent cutter, and by then religating pieces together and performing next generation sequencing, you generate these Hi-C contact maps and can assemble the genome. This is the data, which is, I think, really fascinating that you can make, this is the genome of muntjac revii, which is the 23 chromosome species. And you can assemble basically the 3 chromosomes of the Indian muntjac just truly by telomere-telomere fusions. Without invoking, and that is, I think, the really surprising part of this, without invoking, in this particular case, large amounts of genomic instability that is driven by dicentric chromosome breakage bridge fusion cycles. These are truly linear, continuous chromosome fusions without what we observe in cancer biology, events of chromothripsis or breakage bridge fusion cycles. One of the really interesting part of this study became the analysis of what happens actually to the contacts and the compartments of these chromosomes. If we look at the Hi-C contact maps in the 3 chromosome species, which is highlighted on this part of the diagonal of the contact maps, you see that and this is the 23 chromosome species. You can appreciate that the compartments within a chromosome, the compartments within the chromosome on the Indian muntjac look actually quite different than their counterpart on the 23 chromosome muntjac. If you plot that for one of the chromosomes, the AB compartments that you can generate out of these Hi-C contact maps, you can appreciate that they are actually quite different between the 2 species, which I personally find really surprising, because the inference that one can make out of this is that although these species are highly related, can make an F1 offspring, the individual compartments on these chromosomes are largely different. And of course, the fusions in the 3 chromosome species override many of the AB compartments that are found just by the nature of these being separate chromosomes in the other species. So the implication of this is what does it actually mean that these compartments are present in a cell if they can be completely different in a species that if you were to see these two deer side by side, you would have a hard time distinguishing. Okay, so with that, I showed you that we can detect Hi-C, that we can use Hi-C to detect structural variants that are driven by telomere-telomere fusions. And with this in mind, we now knew that we can use this technology to do to hunt for telomere-telomere events in cancer biology. And so we screened a panel of cancer cell lines with the hypothesis that we would be able to identify telomere-telomere fusions in cancer cell lines. And we did this using the very same approach, using Hi-C sequences of cancer cell lines. And this is a contact map of now a human cell line. You have your 23 chromosomes, and you would see that this is a normal linear depiction of the 23 chromosomes. However, through the screening of the cancer cell lines that we hypothesized might have telomere-telomere fusions, we found one melanoma cell line that turned out to be really interesting using this approach. And that is shown on this side. This is a normal diploid human cell line. In here, you can see that there are many contacts in the melanoma cell line that are off the diagonal, arguing that they are fusion or translocation events of chromosome pieces in this cell line that are abnormal. And if we zoom into this, then you can appreciate that, for example, on this off-diagonal contact, this is a part of the genome where the 12q telomere makes contact to some part of the chromosome 1. So this is a 12q telomere fusion that is somewhere translocated to a part of chromosome 1. And in this particular cell line, we find quite a few of these events. So for example, here we have a chromosome 3 P-arm telomere fusion that makes contacts to 1, 2, 3, 4, 5, 6 to a part of the chromosome 6 in the cell line. And then here we can identify a true telomere-telomere fusion where the 12p arm is making contact to 8q telomere fusion, arguing that this cancer cell and this particular cancer cell that we have identified went through a telomere-driven genomic instability state that can be detected with Hi-C. And so this is just the initial analysis of this particular cell line, but we believe that this is actually a really sensitive assay to detect these events and then to deconvolve how the genomic instability in this cell line occurred. As an outlook, now knowing that we can detect this telomere-telomere-driven genomic instability in cancer cell lines, we are planning to use another of Dovetail's platform, which is called Hi-C, that allows for the assembly of Hi-C contact maps from paraffin-fixed samples. And we're hypothesizing that by taking samples that are undergoing this evolution of cancer evolution, going from a nevus to a melanoma to a metastasis, that we might be able to detect cases where telomere genomic instability is driving large chromosomal events during the transition from a nevus to a melanoma. All right, and with that, let me look at the time. It's almost 9 o'clock. I would like to acknowledge the people that did the work. I mentioned to you the work of Konitoshi Chiba and Franziska Lorber, who worked on the Tert promoter mutations. And then I want to call out the collaboration with Dan Rockshire, who did the muntjac assembly, came up with the initial idea of looking at muntjac chromosomes using Hi-C. Marco, who is my buddy at Dovetail, who helped with all of the Hi-C experiments. Boris Bastian, who provided insight into melanoma biology and the melanoma samples. And with that, I'm going to stop sharing, and I'm happy to take questions.
Terrific. Thank you, Dirk, and thank you to all our speakers today. First question here regarding Cas9. Does the Cas9 destabilization of the Tert locus promote cumulative mutations or instability at this locus as the fibroblasts pass through crisis? And then the second follow-up, I guess, there is, did you also sequence those clones?
Yeah, so so the Cas9 is only transiently expressed in these cells. And so we do not see continuous genomic instability at the Tert locus. And we can confirm that by sequencing of the locus over time. I think that the other question that relates to this that is posted in the panel, our current working hypothesis is that the changes that are required for the additional upregulation for Tert are epigenetic and are not genetic, because the proliferative curve that we see in these Tert promoter mutations doesn't argue that there is an outgrowth of a low frequency genetic event. It, to us, seems much more likely that the Tert promoter mutations functions as a space holder for binding of ATS factors. These are the transcription factors that can bind to Tert promoter mutations. And maybe just this binding event is sufficient to allow for epigenetic wiggle room or inappropriate silencing of the locus, so that over time you can select for cells that have not fully turned Tert off.
Gotcha. Thank you. The next question, just a quick one. Why don't we see Tert promoter mutations in lymphomas compared with other solid tumors like melanoma?
Yeah, so I should have explained that a little bit more carefully. I think the simplest interpretation is that lymphoma probably never fully silences Tert expression, or at least in some cells, that these cells are low-level telomerase positive from the tumor-initiating cell. And so since the tumor-initiating cell is already telomerase expressing, it never undergoes the selective pressure that is required to enrich for Tert promoter mutations. And the Tert promoter mutations is neutral, very similar to the current theory in intestinal cancer, is that the tumor-initiating cell is a stem cell that is sitting in the crib, and that gains that is already in itself telomerase positive. It gains oncogenic stimuli and thereby stabilizes in a Wnt-dependent way the telomerase positivity in that cancer, so that the tumor doesn't require the additional selection for Tert promoter mutations.
Gotcha. Here's going through these questions here. Have you measured the telomerase protein level as a function of time in wild type and TPM mutated cells?
We'd love to do that. Problem here is actually really one of the most fascinating things about telomerase biology. If you look at estimates for how many telomerase molecules are present and are sufficient to immortalize a cell, then this is very, very little. 20 telomerase molecules can immortalize a cell. And that, and on the mRNA level, Tert mRNA level, we estimate that 3 to 5 mRNA molecules are sufficient to immortalize a cell. And so this is, in the field, the hand-wavy argument why we still haven't made a telomerase, a Tert antibody that can detect endogenous levels of Tert expression in these cells. So that's really one of the key challenges, that we do not know Tert protein amount at the endogenous level for lack of detection, because it is so little that we just don't see it. That's why we end up doing this, this radioactivity enzymatic assay, because that gives enough amplification to actually have a quantitative readout of the activity in the cell.
Gotcha, thank you. Are the transcription factors that can bind to mutated Tert promoters expressed before immortalization, or only after a period of telomere-mediated chromosome instability?
Yeah, that's cool. So ETS factors are a family of 26 related transcription factors. Most of them, many of them, are ubiquitously expressed. The factor that is hypothesized or is shown to have probably the largest effect on the expression of expression of a Tert promoter mutation cell is GABP alpha, GABP alpha beta, which is forms a dimer on these Tert promoter mutations, and these are largely, they are ubiquitously expressed. And so although we can't rule out that there are compensatory upregulations on individual ETS factors, it seems that they are bound already at the state where telomeres are still long. They're definitely expressed and most likely also bound to the TPM.
Thank you. Do you think telomere fusions drive whole genome duplication?
Can you say that again, please?
Do you think that telomere fusions can drive whole genome duplication?
Yeah, if I can just say, I believe the amount of evidence that we have for that is limited, but I wouldn't be surprised that it could. The way that that would work is you trigger a DNA damage response, you endo-reduplicate, and you probably just then go through, you skip a division, and then you endo-reduplicate just by the signal that can come from a dysfunctional telomere.
Gotcha. Does Hi-C give you any information on the type of interaction identified, or does it only tell you that two loci are interacting with each other?
Yeah, so that's interesting. Are these true covalent fusions, or is it possible that these are just proximity? We do not know the definite answer to that. It is, to me, it seems extremely likely that these are true fusion events. For the muntjac, of course, we know that they are, because we have physical evidence. I showed the chromosome spread that we know. For the individual cancer cell lines, for the cancer cell line that I showed, we have confirmed some. But yes, it is a looking forward, of course, a concern that we need to keep in mind. That we only see proximity, and we are inferring that they are direct fusion products.
Thank you. we're going to go ahead and wrap it up there. There are several questions that are outstanding. If we did not get to your question today, somebody from Dovetail will reach out directly. If you have any additional questions about Dovetail technology, visit dovetailgenomics.com.
Thank you all. We're actively recruiting. If you're interested in any of the stuff that I showed you today or anything that you find on our website, reach out. We're doing quite a bit of cool stuff, and I hope you all stay safe.
Sounds good. Thank you very much. Again, thank you to Dirk and to all our speakers and for attending today's webinar. Goodbye and take care.